and that given me the opportunity to present this webinar and recent development of our electron microscope facility, maybe India's first cryoplasma-enabled FIB-SEM system, which can be used for both as well as room temperature as well as the cryo samples. So before going, as organizers want that it should be a very basics, and our audience is also MSc students, so I keep it towards the technical. So those who wish to get some very high-end research, which one we have already, but I'm not going to disclose much, but all the examples what I'm going to show in this whole seminar is towards us, what we have generated at our facility. With this, yeah. So this is our facility. It's not changing. Okay. So yeah. So this is our facility I belongs to, and from this recognitions you are have called me for this webinar. This facility is serving the country for last 55 years with more than 2,500 researchers. And because they are getting a complete solutions from sample preparations to electron microscope imaging, so they are using for room temperature cryo-electron microscopy for biological sample. This facility is known for. Services are huge. We are nearly 55 years of the experience about biological transmission electron microscopy with diagnostic quantification, molecular localizations, cryo-electron microscopy, scanning electron microscope, bio-AFM, and confocal with super resolutions, as well as recent plasma 5E system for correlative light and electron microscopy and 3D tomography for lamella preparations. Okay. Moving to this, the facility has pan-India presence. Our users are every states we have presence for the different level are going to south, to northeast, to west, and east directions. Every state has our presentations, and we are giving the services to them. And so its facility is open for anybody to use it. Now, with this introductions of the facility, coming to the basics one, that if any electrons beam collides with your sample, these are the electrons which are going to produce. So mostly the biological people are concerned about the secondary electron. It's going to give you the topographical information. And the secondary electrons are those which is going to produce from your sample. Then the back scattered electron, the same electron is going to be returned back, giving the elemental architecture of your sample. So basics is that this is the electrons which can be used for imaging purpose in scanning electron microscopy. And these are going to be used multiple times in this webinar. So these three, you have to consider the characteristics X-ray, which is elemental detection, secondary electrons, and backscatter electrons. So there are multiple kind of microscope, but many conventional electron microscope we have with us given by the DBT research facility. And these are the imaging, means you can image the surface view. These are the one macrophage cells from COVID patient's lung, from the lung. This is the one nasal epithelial cells, RBCs, and neutrophils. So this kind of 3D imaging you can achieve with the scanning electron microscopy, but it's only the surface view, and depending upon the field view. So this kind of imaging you can generate, but if you have to go the little better one and good one, so now we have equipment for the country has equipment for this cryo environmental scanning electron microscope, which is can we go with 40 degrees centigrade, EDSLA mappings. You can use the hydrated sample for environmental sample. It's have a stem so that we can directly use the grids imaging, at least near to the resolution what we can get from transmission electron microscope. Recently, they have had Trinity detection systems, low wide lenses, mounted VSED for high resolution imaging without sputter coating. So these are the technological development which make current FIV as FIV can be used for 3D imaging. And so if you just see the recent advancement and imaging criteria, so see that the images of the bacteria is going to be very prominent and very well means resoluted. Even then you can see the small vesicles on the surface of these bacteria. These are the some bone marrow cells from the patient samples, and if you can go with the higher resolutions, everything you can detect. Here also you can go with the 3D imaging, but you have a limits for that one, and you cannot get the internal structures. The TM same grid we can view with these ones, and we get it. Fibroblast cells grown on the surface. It's going to be very nicely. You can see the projections and everything by using this environmental mode and cryo-electron microscopy mode with this machine. So this why I'm telling that, okay, though these are the equipment which can add on the advancement of FIB. FIB is not going to be evolved immediately. It's going to evolve with the multiple channels is required. And currently, people have a very good idea that they can generate 3D ultrastructures. Time, there is a one technique called serial sections TM. In the 1950s to 80s, we have to do the manual ultramicrotomy, section it, and then you can get the images and stack it in the 3D imaging. Then later came serial block phase imaging, where you have in-chamber diamond knife. It's called serial block phase imaging. So you have an in-chamber diamond knife. That diamond knife is going to cut your sample. It's a room temperature. And you are going to image it by the same beams. So this is the primary or preliminary of a FIB system, where diamond knife is going to cut or mill your sample, and you can get it still is advantageous because it can go with a very large volume, even the millimeter cube volume you can generate by this SBFI. Then this came array tomography, where the automatic ribbon systems are there, Leica has introduced, and you can get the complete ribbon, image it, and you get the very large volume. Then focused ion beam in 2006, and it has a very good isotopic resolution. I'd say so means it's going to be a resolution imaging small to medium range, up to 100 to means 100 micron maximum. You can go with this one. Then this came a cryoplasma. This is the one a new advantage came in cryoplasma in 2021 with all these 4 lasers. Though it was before also, but all these 4 lasers first equipment came in the market in 2021 with xenon, argon, nitrogen, and oxygens. And it said it can go with a medium to large, very fast than the gallium FIB. And then this is going to be used for 3D with already you know that SP, a single particle analysis, you can do for 3D imaging of macromolecules from 80s they have developing, and after the development of this direct electron detectors, multiple these lenses, it's going to be possible now, along with the multiple development in softwares, we can generate the 3D imaging at the angstrom resolutions by cryo-electron microscopy. And recently it's going to be a cryo-electron tomography, where we have to use high pressure freezing sample because of the tissue, or you can prepare your samples using Vitrobots or Leica GP one-sided blot for cellular system less than 10 nanometer to get in situ structural evaluations after FIB by making lamellas, cryo-lamellas conditions. You can generate nanometer to angstrom resolutions. So these are the very pictorial representations of all these techniques, and it's better for the students who are going to be new in the field that this is serial sections. So you have to make the blocks, cut the blocks in different part and image. Finally, you are going to process the image, and I can, after doing segmentations, you can make the model building and you can generate your image. Then serial TM tomography, again, there's multiple tomography series. You can check it. Block space ACM, where it's going to in column diamond knife, which can means mill the samples, and you are going to get the 3D imaging. And a multi-beam array is also already available, so you can do multiple things, and with directly making this image, you can generate this one. But these all are for plastic resin samples. If you go with the new techniques like focused ion beam, you have multiple options that if the machines is Hydra, Helios, Hydra, or other similar kind, you can use for cryo as well as uh, for the room temperatures. And this one is going to make a trench, and about after making the trench, you can generate the image for your 3D imaging or large volume scope imaging, and finally you can get it. But the same if you are going to use the cryo-FIB-SEM, then you can prepare the lamella. And I'm going to much focus on these aspects, that how to, and after the lamella, you're going to take the cryo tilt series and generate the multiple tilt series. After segmentation, you're going to build your image or model. Or if you want to go with the 3D imaging or volumeoscopy, you can have a slice and view options, or you can have spin milling options for a large area with the room temperature sample, as well as for the cryo-fixed tissue samples or cell sample, and finally you can get it. So these are the currently known techniques where you can generate 3D ultrastructural imaging. And currently it's been FIB, the superior one, that is going to create a very less time, faster, reliable, reproducible, and all kind of imaging you can do with these machines. Even then, you can have integrated fluorescence light microscopy to generate locate the particular area of interest where you want to mill your sample, or you want to prepare your lamella. You can prepare your lamella with fluorescence guided. So if you had to go with the different samples, how we have to proceed for 3D imaging, if you have a biomedical tissues, so you must have to go with the tissue sample with high pressure freezing, because the tissue samples are going to be larger than or thicker than 10 micron, which is not going to be possible to vitrify. I hope that, okay, the audience knows vitrification is meaning that you are going to make a solid water or the ice, which is going to be very means electron transparent. Making a solid ice which can allow the electron beam to pass is the vitrification process for the layman language. So this biomedical tissues we have to vitrify, and for the vitrifications you must by high pressure freezing, because other vitroboard systems is not going to be useful. Then you have to prepare the lamella of that one. Lamella is nothing, a thin slice of your sample, which you maybe 100 to 150 nanometer thick, and you can generate this lamella to get the cryo tilt series. So this lamella you can transfer to your cryo-EM and then cryotomography by using cryotomography tilt series, IMOD, and other software. You can generate your 3D imaging. However, if you have cellular systems, you can have two options. You can go with the high pressure freezing, or you can have just plunge freezing with the single side plot or double side plot. Prepare the lamella, and same things you can generate your image. So these two type of sample, but if you have a cellular or biomedical tissue, you don't want to, yeah. Anything? Okay. So this lamella you can produce with room temperature FIB. Now, room temperature FIB, you can make 3D imaging, slice and view for your resin emitted samples, or spin milling for large area sample. You can produce ultrastructural level details with room temperature as well as cryo samples. So with this, if you have to go, these are the equipment and systems required. For if you are going for the hydrated biospecimen fixations, these are multiple routes. You can go with the high pressure freezing or plunge freezing with GP2. Directly you can go with the semovis or cryo ultramicrotomy, prepare the samples and see, or you can prepare the lamella with the help of a FIB and do all the imaging. Okay, so this is the primary introduction how one could generate the 3D. 5 used for 3D imaging either by slice and view options or preparing the lamella. That lamella can go with the cryo tilt series, cryo-ET tilt series, and that's going to produce the 3D imaging or in situ structural evaluations. So first part of this samples is to produce the vitrified tissues. And if your tissue is thicker, maybe means more than 10 micron, so you have to go with the FIB system, and there is a multiple way to make it. First, you have to cut the small part, do the planchet, make the planchet, and after the planchet, this FIB, this high pressure freezing is going to use. And immediately.
Sorry, Professor. Can everyone please mute themselves? Thank you. Sorry, please go ahead.
Yeah. So this high pressure freezing can be used for making this planchet. The same thing if you have system, you can grow the cell and then you can load on the SPF cartridge. Again, you can high pressure freeze it. Or if you have a cell suspensions, you can go with the copper tubes, high pressure freeze it and use for that one. If you after high pressure freezing, you can use for the room temperature by using AFS2 freeze substitutions and can use for better fixations. Or you can have a cryo fixations you can do by FIB1 with FIB milling, cryo lift out, cryo slice and view. You can prepare the lamella and cryo imaging, or you can do this cryo ultramicrotomy imaging. It's up to you that okay, whatever the applications you want. So this is the one technique that is very simple procedures. A small video is there, but I think it's not. Okay, so this is the high pressure freezing. Totally is going to be used for 10 to 100 micron thickness and must be dialed. Can be used near means live conditions. See that how it's going to be very simple machines. We are going to use it and by just putting this planchet on this system, you are going to just click it, and within a millisecond, this equipment is going to fix your samples. And see that, okay, because of the very, now you can see it's the whole process of high pressure freezing can be used. And if you want to do sectioning before this FIB, SEMO is the one option where you can do a cryo sectioning by Tokiyasu methods or any other method trimming, and you can generate this thin slices for using that sample for your 2D imaging, or even then go for the cryo-electron. This is the one standardized procedure, and we here using for mostly this cryo-immunoelectron microscopy to localize the particular presence of particular proteins by using this immunoelectron microscopy. And you can see that okay, the particular proteins which is near the mitochondrial surface is localized in one corner. So this is going to be very helpful at our facility before the FIB came to us. Before to do the cellular systems, it's better that okay, we can grow the cells, and we have developed this growing conditions. Even then, we can use the carbon copper grid also with the multiple coating. We are able to generate the cells, and mostly people use gold grid. And this whole procedure was standardized at our facility. We can use for imaging, or later we can use for the imaging in cryo conditions after the plunge freezing using GP12. So now, coming to the real equipment, is first in India we have established last year these machines with cryo and multi-plasma focused ion beam ACM. For CLEM and biological FIB-SEMs, what the modes we have here, that you can go with 3D imaging at room temperature at cryo condition, both integrated fluorescence microscopy, because it says IFLM integrated with that one. So it's very easy to localize a particular proteins or particular area you want to prepare the lamella if there is a fluorescence. Uh, Cryolamellar preparations for tomography, you can do with the fluorescence guided one. Correlative light and electron microscopy for 2D and 3D imaging, you can make it. Cryo lift out systems for lamella, you can make it. So you can take a tissue and lift the lamella with half moon. You can use, and it's already, I told you, that's okay, the 4 plasma systems, and it's also had a mode for spin milling, a large area spin milling for room temperature sample. So already just the overview of this machine, how it's work. It's a very basic principle. It's going to mill one particular very thin slices, and the same time it's going to image by the electron beam. So they have 2 beams. One is called plasma, which is either argon, xenon, and nitrogen and oxygen. Anyone you can use for your sample, and depending upon the samples, there is maybe the many combinations are already known, and many combinations are going to come with the time, because it's nascent technology, and with the different sample it's going to be different. But for room temperature, we standardize here that okay, we can make a trench, another thing, by using xenon, and by going for this green clean and imaging, better can do the milling using the oxygen for plastic. But for the cryo samples, we have found that okay, we can make the trench by using xenon, and for the imaging, better is the argon system. So these are the whole systems. That's this machine has IFLM, FIB, ACM, easy lift, and sputter coating as well as GIS coating. So these are the basics units we have with us, and they have a transfer rod with the semi-automatic loading systems. These are the modes with us that we can use here, that anybody can use with high resolution serial imaging. The room temperature, cryo conditions, cryo lift out, as well as cryo lamellar preparation for tomography. For a room temperatures, you can do correlative light and electron microscopy, as well as in the cryo conditions. A large area serial milling we can do with only with room temperature and array. Okay, we can have a 3D slice and view for cryo and room temperature. We have a correlative light and electron microscopy for room temperature as well as in cryo condition, cryo lift out in cryo conditions, lamellar preparation in cryo conditions, and large area serial imaging as well as aretomography. So this is the mode and how we are going to proceed for that one. So it's for the MSc or the first year PhD students who are going to work in these directions. They can notice just I have make it very simple way that how tissues and cells are going to be used. So for 3D imaging, you can start with the block preparations. And then trim the samples, sputter coat it, and transfer the FIB ACM in room temperature, auto slice and view imaging, tomogram generation, and finally you can do a segmentation and model building to get the 3D imaging at ultrastructural resolutions, not the molecular resolution. The same way with the spin milling also, array tomography. So coming to that technical details, how 3D ultrastructure imaging at RT we can do. And we have produced some very good tissue sample. This is the tissue from cervical cancer patient, directly from the human one. And we are searching for the presence of viruses in the late stage. And we could find many samples, which in the literature is known that virus is going to be cleared at the first stage of the cancer, but even in the 4th stage of the cancer, we could found the presence of the viruses in the intercellular spaces. So this is the one first indications applications by using 3D imaging, you can get it. So the same TEM block we are using, whatever is going to prepare for our own samples to image. After ultramicrotomy, we are going to just stain it. Uh, just transfer on the ACM stub, and this stub is going to transfer to the FIB, and finally we could generate this 3D imaging. However, in the normal TM route, you have to go with the cells, plastic resin, ultra sectionate, thin slice of 70 nanometer, and finally image it. So this is going to give a very good informations, 3D informations about the cells. And we have developed many 3D tomogram of cervical cancer and different kind of the tissue using this technology. So just see that's how even then this the whole methods how we are going to first you have to make the gold sputtering, transfer it, and first you have to make a trench. And this is the area of interest where we are going to image the 3D one. So you can generate all this trench, and finally, after the green clean, you can do slice and view and generate the 3D tomogram of this one. And this is the tomograms already we have generated at our facility. And you can segment it very easily. You can segment the nucleus and other things very nicely. You can see this segmented volume of this image. So see, at the initial point, this is the one when we are using Xenon as trenching imaging part, or for ACM imaging, see, this is the curtain lines are going to become. So this is we learned that, okay, by changing the just xenon to oxygen, this sample is going to be much better. Now you can see this. This is the same tissues. You can see the same one cell having, even then you can count the mitochondria, which is white nucleus, nucleolus, all these chromatin materials. The complete details, even in the vesicles present, extracellular vesicles present in the cellular system. So this way you can generate, this is the primary beauty of this 3D slice and view. Even then some filamentous structures, Golgi complex, everything you can visualize, and after the segmentation, the same image is going to be better. So this is the primary usage, immediate usage of this FIB in biomedical field. You can get a biological field for getting the better information, which is not possible by simple EM, or maybe that information is missed because of the simple one slide image, which is going to be very clear in the 3D imaging. We have also large area volume systems, and we can do all this one by using just changing this stage. And you can generate the large area volume by spin milling a single cells. We can generate, and finally you can generate the tomogram. So if you see that this is the one our plastic resin sample, and these are the trench which one we have used for slice and view. And this is the one trench we can use for spin milling. So the area, this is the maximum 100 micron area we can use it for spin milling, but for slice and view, by ASV, auto slice and view, and this is for the spin milling. So large area can generate the large area image, though the resolution is poor, but it's going to be improved with the time. And just see that, okay, how it's going to show the results with the yeast cell with conditions. Now I can see this is the one large area volume samples, and multiple cells together we can see by using spin milling of the samples. This is the one another large area samples by spin milling. You can get it, and you see that, okay, this is the same again, the cervical cancer fourth stage tissue, and these are the some outermost cells, which is dead. And if you go in internal cells, you can identify each and every cells. It's macrophages or other cells, nucleus, and other presence. Now coming to the cryo mode of FIB-SEM, we can have a similar way that, okay, you can go 3D or high pressure freezing by HP transfer in a 5B auto slice bioimaging tomogram generations. But for the cells, you have to first go the cells on the grid or just pour the drop cast the grid cells on the grid, vitrified by plunge freezing and transfer. So many information you can get it by this one. Many literatures is already there where people are using the cells for tomogram generations and small organisms, and the information is a little less when the tissue is used for auto slice and view. Similar way, we can prepare the lamella. It's a primary use of this cryo conditions, lamella preparations by using autotem. Everything is standardized. And very simple systems there already. This softwares of this machine is set, and you can any persons knew the whole process. You can generate the data at lamella preparations at this facility. Then lift out is a little tricky. As well as correlative light scanning, light and electron microscopy, you can use with cryo lamella preparation and correlative light and electron microscopy. Coming to that just detail, how it's going to be useful for that. So cryo grid preparations, again, you have to just grid, plunge vitrified it by plunge freezing, all this special kind of this searings you have to use where there is a notch for that one, and multiple companies are making different kind of notch. Finally, you have to transfer in the shuttle, and shuttle is going to transfer in cryo conditions to a FIB-SEM. And you can generate by after multiple sputtering means first layer of sputtering, second layer of GIS, then third layer of sputtering. So initially we have done and we tried to get it the images, but it's very poor quality at the time when we have started in this year January about getting this data. So these are the some data, so you can see the cells, but it's not sputtering, not very proper. See that the one grid we have used, and this part is the one where we are going to use for auto slice and view in cryo conditions. These are the yeast cells on the grid. We have milled make a trench for auto slice and view options. So now it's going to be more clear for you. Slowly it's going to be improved, and the other part we are used for the lamella. But going to this, you have to make some fiducials so that it can be localized, and whole process you can generate it. So the first image was very poor quality. So because I have to tell ifs and buts for the students, so you have to just feel it. And now here we can feel it, but later we can move it with after the standardizations and see the cells. Each cells completely we could see in these conditions. The next tomograms we have generated for that one, little ice contaminations are there. We are standardizing this one. But see this, the whole cells, even the dividing cells and others, you could easily identify, and the tomogram can be generated by this one. From a slice and view of each cell, which is plunge freezed by using. So now coming to the cryo slice and view of HPF tissue samples, we have tried last 1.5 months of this technology, that how we are going to see this initial phase. We could not get anything because of this sputtering, and GIS layer is not proper. And this is very important to get the good imaging. This layer is very, very important to make it once you are going to put the when it's sputter, then GIS, then sputtering. So 5-layer GIS and sputtering, we have done it. And each sample to sample is need to be standardized. Later we can get little better image, but still here you can see the second sputter line is not going to be very good. It's a very uneven and not smooth. So again, we are not going to get the very good data. Afterward, the next we can get standardize the conditions, and now you could see that complete layer of GIS as well as this is visible, and this outer layer, upper layer is finally sputtering layer, and you could see the some information. Still, we are standardizing this technique that okay, after the high pressure freezing of a tissue, can we do slice and view as well as prepare the lamella for cryo lift out. So this another thing, how to generate the cryo on grid lamella preparations. You can make it, the process is cells on the EM grid. You can generate the atlas, sputter and GIS coating. Finally, you can milling and thinning. You can do and can produce the lamella. And finally, this lamella is going to be cryo ET till series data collections. You have tomogram by IMART, and finally you can have segmentations. You can segment these ones and you can get it the very nice image alive. The one blue-green algae, we can get it. We have made it the sample for that. The lamella preparation, see, first you have to make the trench, and then we are going to finalize the mill both area. So this is the one, it's a thicker lamella. We have to make thinner by just thinning multiple way. And once this lamella is going to be prepared, you can either lift out or like the same lamella you can use for imaging or cryo tilt series. Cryo tilt series or cryo electron tomography tilt series generations to get the 3D imaging. So first that's why the FIB view, now this the ACM view of the lamella, the same. You could see this, we have prepared the, now with the multiple thinning, slowly it's going to thin, and finally the thin lamella is produced. So this is the one lamella. Later we gone for a little more thinning, and finally this could the lamella produce with multiple samples. So multiple view and multiple angle view, you can see the different one. So this is the one of the first lamella we have prepared. We could say that, okay, the first lamella might be prepared and in the cryo condition using the yeast system. So later we can produce many others one. Now you can see this how to do cryo on-grid lamella preparation with the CLEM, the cells and EM grid. Again, the same procedure. Here you are going to use IFLM to localize your particular area of interest where you can generate the lamella for that one. And see here, these are all visibles at our lamella conditions. The same samples, how to do the correlative light and electron microscopy. We have another slice and view options. So see how it looks like under this light microscopy or DIC image. And finally, once we are going to see and merge the both images, you could see that, okay, this is our area of interest where we can generate the data. So this is very good, helpful to do cryo correlative light and electron microscopy directly from one equipment. When same sample, same equipment, it's not like that we are taking the different image of EM and light microscopy separately and merge it. Here the same equipment, same sample together simultaneously, you can generate this cryo light and electron microscopy, which is also going to be a very less lab, very few lab in India they are doing for this kind of experiment. So finally, we could, by the lamella preparations, we have generated some tomograms of the samples using this cryo-electron tomography of our one. So in summary, if I have to summarize that, okay, how this FIB is going to be useful for 3D imaging, ultrastructural imaging, lamella preparation, and cryo correlative light and electron microscopy. That you have a sample, you can trim it, and finally you can make this technology, 3D imaging. You have which can generate it. So all these, but best part is that, okay, you can take a 5E1, can generate serial plasma slice and view, you can take it, or you can do cryoplasma FIB systems by making this lamella and produce the cryo-TM 3D reconstruction. Similarly, you can do IFLM cryo slice and view using cryoplasma FIB ACMs and can generate the lamella. You could guided the lamella preparations, and finally you can generate the very good nice 3D tomogram for your samples, which can be used for cryo in situ structural elucidations of the sample. With this, I thanks DVT Sahaj facility, who has very generous enough to fund for all the equipment for sample preparations as well as a 5 to our facility. With 200 kV side entry microscope, we could generate this data for the electron microscopy and AIIMS, who has given the opportunity facility to create the such facility. Thank you very much, and I'm open for the questions.
Thank you, Professor Subhash. That was very, we learned a lot from the talk. So this is a very new technology, and it is, as we know, AIIMS Delhi is pioneering it in India. So if the students have any questions, they can raise their hands, participants. Or if you would like to type out your question in the chat, we can also read it for you.
Yeah, if you have not understood, just let me know every slide. I can go back the slides again.
Yeah.
And many things together. I have just given the overview. So
yes.
That, what we have to get.
Yes. Yeah. No, I think it was very nice overview of everything. So are all of the facilities available now for use to the community?
Yeah, it's open.
Okay.
It's open for this one. Mostly for cryo lamella preparations and slice and view for plunge freeze sample is open. For room temperature means this slice and view is open. Only we are struggling for high pressure freezed tissue sample to get it, because some problems we figure out that okay, it's a problem might be with our with high pressure freezing, not with the equipment. So we were just sorting out how to sort it that.
Okay. Okay, so I think we have one question. So from Nandini, I'll maybe just read out the question. Would you recommend adjusting the GIS layerer sputtering coating to reduce artifacts during cryogenic slice and view?
Yeah, but you have to see that okay, what type of if you are going to be much thicker layer, we also get the problem. I mean, this one that when you are going to use this surfactant also, we get a one surfactant layer also on that one. So it's depend upon which is on surface, the area which one you are going to choose. If it's very thick lipids, then it's better you have to just do some pre-preparations to remove these lipids if you are not interested on the lipid parts. So, and then you can go to high pressure freezing or plunge freezing and make the sample ready. So I think this way, it's already, yeah, it's a tricky situations. It's depend upon your samples and stability, what type of samples you want to do. So. Yeah.
Hi, Ashwini. Yeah, please go ahead.
Hi, Mamta and Subhash. Thank you very much for such a wonderful presentation.
Thank you.
So I was wondering whether any progress on the plant issue.
Yeah, you have to submit the tissue, then we save the data, because we don't have much plant tissue. I'm depend upon the user for the plant tissue. So.
Right. So we will be in touch, and then maybe one day I will visit there and see that.
Yeah, you can.
The data on the east looks promising, right? East data.
Yeah, east is also means even then that also you can have means if you have a blocks, you can just submit the same block. You have suppose that you have taken some blocks of a room temperature. First, go with the room temperature slice and view and see that okay, you are getting your answer or not. If you are getting your answer, then you can go with the cryo conditions, and it's better. I am suggesting everybody that okay, if you have a room temperature blocks ready with us, the same block we are using. All these ones we have not did any new things. It's older blocks which one used for TEM. We standardize this technology for our machines for our samples, and we are getting the data.
Right.
And very nice images you can see that now. So.
Right. Okay. So okay. Thank you. Yeah.
Thank you.
I think one issue that is needed for visualization, at least the initial stages,
I mean, it's for room temperature. They want this one room temperature sample slice and view for slice and view or for lamellar preparations.
So I think this is a continuation of the previous question with respect to lipid-rich tumor tissue. Cryo.
Yeah, so cryo maximum you can go with a 200 micron. So I suggest that your samples maybe it's better that once you are going to use cryotomes, section is a very thin 100 micron sections, and that section you are going to use. It's going to be better result in high pressure freezing, flat surfaces, everything is there. But if you are going to use any small sample, if more than 200 micron thick, it's a little bit that's going to be very good vitrified. There is a ice contamination on the surface, and that's why they're creating a lot of problems for us. So better use cryotome quickly and take that thin slice. Immediately you can plunge freeze it or high pressure freeze it, whatever you want. But plunge freezing you need a very thin sample, but it's very difficult to use it for that. Yeah.
Okay. Hi, Manoj, go ahead.
Hi, Mamta. Great talk, Professor Subhash. So I was wondering about access. So what is the process for access for people who are not associated with any university or in a private university?
Yeah, it's open for everyone. So we have a total 3-4 categories of the users: universities, research institutions, and private. So anybody can, if not associated, they can apply as a private one. And it's open for everyone. Anyone can use it. It's not that you should be academician, but yeah, there is subsidies for academicians and researchers than the private and profit-making institutions.
I have a follow-up if I can.
Yeah.
So I was wondering about, processing and compute. Is it available, or is it like you can give remote access to people who collect data and they can process, or they have to be present at the center?
Best part is if the users present. This is our best practices here. That's okay. At the time of imaging, we assume that users should be there because he is the one master of his own samples. It's not like a simple technique like SPA, single particle analysis. Anybody can generate the. If you are not able to come, we have some video systems that can show you the screen, and once you're going to tell that this area I want to do it. So this area marking is the primary important for this one. If you're going to do slice and view, or if cellular systems are there, then we can make it at our own. So it's both ways possible. You can come, or you can have a remote access. Yeah, once the data will be ready, we can give you the remote access you can download the data from our own. So we are developing the whole server system. Now every server is connected with each machines. Data is poured there, and we can give you the access. So for 2 days or 3 days, you can download your data. Or if you want to compute, we don't have a much GPU-based network, but we have 3 high-end so you can process the data if you are going to be here. So,
thank you.
Excuse me. Hello, can I have a question? Time is available.
Yeah. Please, Shubrata, go ahead.
Yeah. So I have one technical question. Let's say you imagine one situation where you are inserting some molecules to the cell. So it's going to the cell. Now inside cell, it is making microstructure. And that microstructure is luminescence. So, okay. So now my question is that by microtome, is it possible to say that microstructure where it is located within the cell? So that kind of, what is the advancement of this microtome? Yeah.
It's very. This is the one. At least I think you have seen the data. We found patch of 5 to 10 means viruses. So till means we want to show the large area. If you want to go with only one cell view, then it's going to be very large field of view, and you can get this kind of means currently with the room temperature slice and view, we could very easily see the structure which is less than means 10 to 15 nanometers. If your patches is 10 to 15 nanometer, room temperature slice and view works. If it's smaller than that one, then you have to go with the cryo conditions, and there you can use fluorescence guided one, and you can easily locate the particular area where it is coming from. Or you can prepare the lamella for that part and go with the detailed structures. But if you want inside where it's whole cell, complete one cell and where your molecules.
Yes. Yes.
So
Yes.
then you have to go with the slice and view in cryo condition. And if it's larger in size, better use room temperature slice and view first, because the resolutions, contrast, everything is better here. Directly you can image it. But for doing the cryo condition, you have to do tomogram generation, everything, and then finally you could see that particular molecule.
Yeah. Okay. Thank you, sir.
Any questions? You can also ask about how these different techniques are done as well, if anybody has any questions or doubts. Nandini, do you have a question? Your hand is raised. Okay. So the question that Nandini is asking is, could correlating the HPV viral load with the ultrastructure help identify biomarkers for early stage detection before the tissue abnormalities occur?
See, if you go to the disease biology of cervical cancer, so it started with the viral infections. So it's very easy. Means you don't need to go at such a high-end technique to find out the viral load. But if something is going to be there, suppose that they are going to produce some oncogene like E7, E6, and that you want to correlate, then that time you have to use some kind of correlative like microscopy along with the means nowadays a lot of new antibodies fragments are there which can penetrate the cell, which can go in the live cell, and that is going to be used for both. They have fluorescence as well as gold coating, though it's very costly. They are selling it, but it can be used for that one. But I don't understand the clear questions. Why? What is the purpose of what the biological question you are going to answer by using this one, sir? It's already known. Simple immunofluorescence can tell you the data that okay, the virus is there inside with confocal. Why to go to this high-end techniques?
I think she has a follow-up question. Any precursor assays that would be better?
For what? Means to identify early detections.
I'm guessing that, yes.
Yeah, so see, it's a practical one. Early detections problem is to getting the sample, because it's started with one cell, and sample collections or cytology sample, you diagnose as a non-cancerous, but because the cells number is going to be very small, it's create the problem only after scene 3 on other stage, all this PEP smears or immunofluorescence, or even then histopathology can tell you, because the getting the samples right place is again the problem. It's a medical problem. So it's not our microscopic problem. Means they have to collect the right data, right samples of that. But yeah, early biomarkers is possible if you have to come with some new early marker. Till now, there is no early markers are there. Only the viral infection is the one. They are going to see the viral genomes by RT-PCR, and later they are going to use the immunofluorescence or histopathology for that. Pipe smear imaging.
Okay, I think, yeah, if there are no further questions, I think we can thank Professor Subhash. Yeah, thank you for giving us an overview and introduction. I think as the community does more of these experiments, we'll have more to discuss and we'll have more communication.
Yeah. But this one's it's going to be, and we are open. Anytime they can write the questions, we will answer them. So,
oh, great. Yeah, that's good to know. Yeah,
anybody can visit also. It's open. It's not a closed system. It's a public system. So there's no need to get any permission. Directly, anybody can come to the facility. Yeah.
Very nice to know. Thank you. Thank you for that.
Thank you.
Thank you. Thank you very much.
Thank you. Thank you, Professor.
The listeners are going to be benefited with some technical talks. So, we'll see.
Thank you. Thank you very much. Yeah.